Clostridium perfringens is one of the most widespread disease-causing microorganisms in nature. It is primarily found in soil and in animal manure. From here it can be transferred to several foods, e.g. vegetables, meat, poultry, and fish, as well as spices. The bacteria can also be found in contaminated water, including contaminated drinking water. Clostridium perfringens is a gram-positive rod-formed bacterium. C.L. perfringens is a spore-forming bacterium that is often found in low numbers. There are often only a few bacteria per grams of food. A spore is a kind of resting stage for the bacterium, used for survival in extreme conditions. Spores can survive extremely high or low temperatures or total desiccation, which bacteria normally cannot. Under these conditions, the bacterium will therefore die, while the spore will remain. If the growth conditions become good again, the spore will be brought to life and the bacteria will continue to grow. In the case of poor manufacturing hygiene or incorrect storage, the bacteria can be present in high numbers, which can cause food poisoning. Clostridium perfringens is quite resistant to water treatment processes, and controlling the drinking water is conducted to ensure that it is not contaminated. Clostridium perfringens can grow within the temperature range 12 to 50 degrees Celsius. It can grow very quickly, as the number of bacteria can double in less than 10 minutes. The bacteria's spores are very heat-resistant and can survive boiling. The spores can often be present in salted and dried products where oxygen is absent. Clostridium perfringens will not grow in the presence of oxygen, as it is obligate anaerobe. Clostridium perfringens can grow in foods that have a high salt content, up to 5%. Vacuum packing the food does not eliminate C.L. perfringens, but packaging in a modified atmosphere will in some cases inhibit its growth. Clostridium perfringens can grow in foods with a pH above 5 and below 9. Food poisoning caused by Clostridium perfringens results from slow cooling of heat-treated food combined with insufficient reheating before eating. The poisoning is caused by a toxin produced by C.L. perfringens in the small intestine. The symptoms are diarrhea and abdominal pain, often combined with nausea and vomiting. It usually starts 8 to 12 hours after eating infected food but can take from 6 to 48 hours. The symptoms usually last about a day, but diarrhea can occur for up to 2 weeks. Complications are rarely seen. To detect Clostridium perfringens in water, you must use 2 to 3 eggs, TSC medium, 70% ethanol, water, colony counter or light table, sterile tweezers, sterile 100 ml measuring cylinders, sterile pipette tips for 2 to 10 ml pipette, and 200 to 1000 microliter pipette. Sterile 0.45 micrometer nitrocellulose filters, sterile petri dishes, sterile filtration units, a standard laboratory balance, sterile containers for egg yolk emulsion, incubator for anaerobic cultivation, oxygen removal bag for anaerobic cultivation, sterile 0.85% NaCl solution, 37 degrees Celsius incubator, and a 45 degrees Celsius water bath, vacuum flask, vacuum hose, and vacuum pump, 200 milligrams of cycloserine and sterile water. To grow Clostridium perfringens in the laboratory, the so-called TSC medium is used. TSC stands for tryptosulfite cycloserine. Tryptose is broken down protein which consists of amino acids and peptides and, together with yeast extract and soy peptone, constitutes a source of carbohydrates and amino acids for the bacteria. Sodium metabisulfite and iron ammonium citrate are used as an indicators of sulfite reduction. Clostridium perfringens carries out sulfite reduction and thereby produces hydrogen sulfide. Hydrogen sulfide reacts with iron in the medium, forming iron sulfide, which is black. TSC media is also available premixed. Here, only water must be added, mixed, and the pH value checked. After autoclaving, the still liquid medium is placed in a water bath at 45 degrees Celsius. The medium must not solidify and be reheated. Egg yolk emulsion is prepared by placing 2 to 3 eggs in a beaker with 70% ethanol. The eggs must be completely covered by ethanol. Leave the eggs for an hour. The eggs are then washed with a stiff brush and washing up liquid, and the eggs are separated into yolk and white. Use autoclaved beakers for the two fractions. Prick the yolks so that they float out with a sterile scalpel or a sterile disposable pipette. Transfer the egg yolk mass to a sterile 100 milliliter measuring cylinder. Measure the volume and pour in the same volume of 0.85% sterile sodium chloride. Mix carefully, possibly using a sterile stirring magnet. Add 12.5 milliliters of egg yolk emulsion to 250 milliliters of TSC medium using sterile pipette tips and aseptic technique. Medium with added egg yolk emulsion is again placed in the 45 degrees Celsius water bath. Add 2 milliliters of sterile water to the glass with cycloserine. Mix well and add 1 milliliter of dissolved cycloserine to 250 milliliters of TSC medium using sterile pipette tips and aseptic technique. Mix well the medium with added cycloserine and place it again in the 45 degrees Celsius water bath. 15 to 20 milliliters of TSC medium are poured into sterile Petri dishes using aseptic technique. The Petri dishes are left for 15 to 20 minutes until the medium has solidified. They are then labeled with the date, name of the medium, sample name or number, and the pH value of the medium. The lower part of the sterilized filtration unit is placed on a sterile vacuum flask. Using sterile forceps, place a sterile 0.45 micrometer filter into the filtration unit. Attach the upper part of the filter unit. The ice sample must be thawed before 100 milliliters is measured with the sterile measuring cylinder. The water sample is mixed and 100 milliliters measured using the same procedure. Vacuum is applied and the sample is poured onto the filter, avoiding splashing up the sides of the filtration unit. When the sample is filtered, the vacuum hose is removed from the vacuum flask and the filtration unit is disassembled. With sterile forceps, transfer the filter to the agar plate with TSC medium. It is important that no air bubbles accumulate between the agar and the filter. With change of sample, remember to use a new filtration unit, a new 100 milliliters measuring cylinder, and new sterilized tweezers. The plates with filters are placed in the metal holder belonging to the anaerobic culture container. The base must face downwards. Lubricate the rubber gasket in the lid of the anaerobic culture container with silicone oil if necessary. An oxygen removal bag is opened and within 1 minute, the oxygen removal bag is placed in the culture container and the lid is clicked on, being careful to check that the lid and container are tightly closed. The culture container is placed in an incubator at 37 degrees Celsius for 24 hours. After the incubation, growth of black colonies is seen on the TSC plates. The smell of hydrogen sulfide is evident when you open the anaerobic container. The black bacterial colonies can be Clostridium perfringens, but they can also be other bacteria that can produce hydrogen sulfide and that can grow both with and without oxygen. To test this, a sample is taken from all bacterial colonies which are plated out on 2 ordinary PCA medium plates with neutral pH. The plates are incubated at 37 degrees Celsius. One plate is incubated under aerobic conditions, and the other plate is incubated under anaerobic conditions. Clostridium perfringens cannot grow in the presence of oxygen. Therefore, no growth is expected on the PCA plate incubated under aerobic conditions. To test for lack of motility, a sample is taken of each colony on the filter and inoculated onto semi-solid agar. Clostridium perfringens is non-motile and will only grow close to where the sterile inoculation loop has been inserted. We have used a modified RCM with 0.5% agar to check for motility. As a control, a motile bacteria is also incubated. This bacteria will move, and the medium will become opaque. To check for strictly anaerobic growth, we have also prepared a liquid RCM medium. The combination of lack of growth on PCA agar with available oxygen and lack of motility in semi-solid agar indicates presumed Clostridium perfringens. If the bacterium grows on the PCA plate under aerobic conditions, It is not Clostridium perfringens. Several black colonies from the filter showed lack of mobility on semi-solid RCM medium. These colonies also showed lack of growth on PCA plates incubated under aerobic conditions, and in the liquid RCM medium, there was only growth at the bottom of the tube, where no oxygen is present. This strongly suggests that we have a non-motile bacteria that is strictly anaerobic. In the semi-solid agar, large bubbles appeared, a clear indication of gas production. The content had an unmistakable smell of hydrogen sulfide. In connection with this analysis, a gram stain of suspected Clostridium perfringens bacterial colonies was performed. Not all the bacteria showed clear gram-positive staining, but bacterial spores were present in the preparation.